I'm going to be reading again from my book, Mind and Evolution, as assessed through reviews of major texts. Junk DNA, a journey through the dark matter of the genome, by Nessa Carey, 2015. Seriously? Or is Nessa Carey kidding? Can so much have been discovered in a field so recently established, involving such improbable scientific achievements? Well, the book is published by Columbia University Press, has 52 pages of notes, index, and listings of human diseases cited in the text. And Nessa Carey is a former senior lecturer in molecular biology at Imperial College London. So I opted for taking her seriously. If she is serious, then we have to rebuild the discourse we employ for discussing evolution from scratch. She's revealing details of what we have to account for. Ready? My extracts from her book could almost fill a book by themselves. I will limit myself to how junk DNA influences human evolution and the human mind. Here's the gene-centered account of the genome that most of us currently have, on which we've based our sense of how evolution works. It emphasizes how stable the genomes have been over time. Quote, over 98% of the DNA in the human cell is junk. It doesn't code for any proteins. In terms of numbers of genes that code for proteins, humans contain pretty much the same quantity, around 20,000, as simple microscopic worms. Even more remarkably, most of the genes in those worms have directly equivalent genes in humans. But take a junk-centered view, and what's notable is how unstable the genome is. Quote, you would predict that there should be a significant degree of similarity between the human genome and, at the very least, the genomes of other mammals. The problem is that only about 5% of the human genome is conserved across the mammalian class, and the conserved regions are highly biased towards the protein coding entities. Okay, so isn't that what you'd expect if the non-protein coding sections of the genome really are just junk? But latest estimate is 80% of the human genome has function. And if it is confirmation of human exceptionalism you want, our genome's junk is where you'll find it. Here's why we're special. Quote, why are humans such complicated organisms if our protein coding genes are similar to those from flies and worms? The more sophisticated an organism, the higher percentage of junk DNA it contains. The more complex an organism becomes, the more the genome begins to resemble Lego, which to a much greater degree of flexibility in how the components are used. End of quote. One measure of this flexibility is how many ways what we think of as individual genes can be cut, spliced into different protein coding DNA and RNA segments. Again, we humans seem privileged in this respect. Quote, human cells are able to generate a greater degree of protein variance from one gene than simpler organisms. Over 60% of human genes generate multiple splicing variants. End of quote. One kind of junk, long non-coding RNAs, I'll repeat that, long non-coding RNAs, appear to be particularly sensitive to evolutionary pressures. Quote, there are lots of them, although no agreement yet on the precise numbers. Estimates range from 10,000 to 32,000 in the human genome. Only about 12% of the same class of non-coding RNAs that are found in humans and mice can be detected elsewhere in the animal kingdom. Only 2,500 were conserved across tetrapods, of which a mere 400 were classed as ancient, by which the authors meant they had originated over 300 million years ago, around the time when amphibians and other tetrapods diverged." End of quote. As for what these non-coding RNAs can do, consider their apparent role in the brain. Long quote. "More long-coding RNAs are expressed in the brain than in any other tissue, with the possible exception of the testes. Many of the non-coding RNAs expressed in the brain are specific to humans or primates, and this has led researchers to wonder if they could be responsible, at least in part, for the hugely complex cognitive and behavioral functions found in higher primates. Different types of long-coding RNAs are expressed if we examine samples from different brain regions. Enthusiasts for long non-coding RNAs claim this supports their model for the importance of these molecules, because why else would different brain regions switch on different long non-coding RNAs? A long non-coding RNA has been identified that influences how the cells in the brain form connections with each other. Another long non-coding RNA, which has evolved since we diverged from the other great apes, may be involved in regulating a gene that is required for the unique development processes that generate the human cortex. The examples above suggest that long non-coding RNAs play beneficial roles in the brain." End of quote. Epigenetic mechanisms are particularly evolved in gene regulation. By turning on stem cells in the brain, they can contribute over our lifetimes to development of what you might call wisdom. She gives an example I'll read that again. Epigenetic mechanisms are particularly involved in gene regulation. By turning on stem cells in the brain, they can contribute over our lifetime to development of what you might call wisdom. She gives an example coming out of laboratory experiments on mice. Quote, "Although we usually think of our brains as being fully formed in adults, recent data has shown that even in this organ there are some stem cells. In animals that rely on a highly developed sense of smell, these stem cells can be activated to form neurons that respond to new scents. This allows the animal to tailor the smells to which it responds most strongly." End of quote. Carey mentions several ways epigenetics may be what changes our mind in ordinary life. One example is by methylation. Quote, I'm going to read that again. Carey mentions several ways epigenetics may be what changes our mind in ordinary life. One example is by methylation. Quote, "The DNA in our cells is wrapped around bundles of 8 histone proteins. Often our cells need to respond to short-term changes in the environment. They add modifications to the histone proteins adjacent to genes. Changing the histone modification can turn genes off, but because these modifications are relatively easy to remove, the cell has the option of turning the genes back on fairly quickly if it needs to. The histone modifications can also be used to modulate the expression of a gene. Turn it on a little, quite a bit." End of quote. Here's a summary of epigenetic influence. Quote, one stretch of DNA can include a protein-coding gene, long non-coding RNAs, small RNAs, antisense RNAs, splice signal sites, untranslated regions, promoters, and enhancers. One stretch of DNA can contain all those in that one stretch. Layer onto this the effects of variation in DNA sequences between individuals, changeable three-dimensional interactions, plus binding to other RNAs and proteins, then add in the effect of our constantly changing environment. End of quote. Carey spent more than a decade as senior director at Pfizer consulting on epigenetics. Besides describing epigenetic mechanisms in detail, she also shows how study of them is illuminating medicine. But what served my interest in the evolution of mind was how many, varied, and complex the mechanisms of epigenetics were, and how enormously rich the life of the genome must be. It resembles that of a king, with an entire court of functionaries serving his interests, and many departments to carry out affairs of state. Carey forces on us the question, how is this elaborate court run? How could it have evolved? Does it constitute a mind? If not, what is a mind?